and welcome to the afternoon session. I would like to start by plugging our next panel, which will happen at 14:45. It's our big pharma panel, and it will be great if as many of you would like to stay for that as possible. But now I'd like to plug Lumito and CEO Mattias Lundin. Welcome, Mattias.
Thank you so much. Thank you. And great to see so many people here. This is the first presentation after lunch, usually a challenge, but I will try to keep you awake, I promise. I'm Mattias Lundin. I'm from Lumito. And Lumito is a medtech company within diagnostics, digital pathology company. It has its background from the university here in Lund, from the famous Department of Physics. We are providing or making digital images based on up-converting nanoparticles, so laser and nanoparticles in combination on tissue. That's what we do. And the product development is now finished, so the product is there. And now we are in the compliance testing phase before the CE mark. A little bit of background. The company was founded in 2010. But the current journey that we are pursuing really started late 2017, where the actual product development started. And then we've been pursuing that for the years in the past, and here we are now with the final product. Over the past 2 to 3 years, we've been doing a lot of studies, proof of concept, external pre-studies and internal studies with pathologists and researchers in Sweden. And now lately, we also finalized the very first study together with a research group in UK within renal diagnosis, renal biopsies, with promising results there as well. The very last, or the latest, I would say, milestone that we passed was manufacturing of the reagents that we need in order to complete our product. So our production partner, Nygen in Finland, they finalized the manufacturing of the commercial version of the reagent just a couple of weeks ago. So what about the product and some more details around it? Yeah, it is about a digital image, a very contrastful and precise image. And in order to get that, we are using a scanner with LED light, bright field light, and laser. And we also have to the left here, nanoparticle in those bottles, in that small bottle, that you are applying on the tissue sample. And then you put it into the scanner and making the images, very, very precise images. The product advantages that we have is from the left, providing a high contrast and granular images more than you can find currently in the market. The physics around the technology avoids completely autofluorescence. And this is a well-known challenge within diagnosis and analysis of tissue using IHC. The product has a very low detection threshold. So meaning that if there is anything positive linked to the biomarker, it will be detected with the technology. Finally, to the far right, we are able to combine bright field, traditional way of looking at images, combining with the new way, the unique way, Lumito through the UCNP, the up-converted nanoparticle technique. We are able to combine those two technologies. Here you're having an example, is a breast cancer tissue. It's labeled with DAB, so immunohistochemistry, that's the brown part, which is the biomarker HER2. And in combination there, the green dots that you see, it's a progesterone as a PR labeling, and that's the UCNP. So we are able to combine these two worlds, the traditional way of showing it, and then showing in combination. So this is a multiplexing using chromogens and the UCNP marker. The market that we are addressing, that we are entering, is the market of tissue diagnostics. And it's a huge market. It corresponds to $5.3 billion annually. It has a projected growth for the next 4 years on 7%, and it's mainly driven by the global aging population. Within tissue diagnostics market, you have an area called digital pathology, and that's the area where we are entering. That has a projection of stronger growth. In the next 4 years is projected to grow 13%. The players in this market, the leaders within tissue diagnostics, are Roche Diagnostics, Leica Biosystems, part of Danaher Group, and Thermo Fisher Scientific. And we've been selecting the research market as the first entrance, and then the clinical histopathology market as the second entrance. If we're then cooking it down to the more immediate market to be addressed, which is the European research market. And it's a decent volume on almost $350 million. And that's the market where we will launch our very first product. And the customers here, the largest part is pharmaceutical companies. It's contract research organizations, and it's also academia and forensic labs that it's adding up to this volume. Our solution fits perfectly well into the standard workflow in IHC. So they don't have to change anything. So it's just following the procedure that are used in laboratories across the globe, which is very convenient and good, I have to say. We protect ourselves and the technology that's been invented here in Lund through 3 patent families, and we are covering the key markets that we believe are important. In addition to that, we are filing more patent applications, and this is a way that this is a work that we continuously do. The mission that we have is to become a global leader within digital pathology and constantly come up with new solutions for the future. So innovation and be a step ahead of the game, it's very important for us. What we do currently with the current portfolio is to provide a digital pathology solution, including an IHC kit. And we would like to do that to help laboratories to free up resources, getting more information from each and every slide, and increasing precision. Being more precise is good, and minimizing thereby human errors. We aim for global reach, but are selling direct in the near market like Sweden and Scandinavia. Innovation is important in our core technology, UCMP, and image analysis. We have selected to have a scalable production to outsource the production. So in short, what do? We are aiming to get the CE mark in place and launch, of course. That's the most important thing. We are already now pre-selling the solution, and we are maintaining contacts with the global leaders for future collaborations. In the long term, after we have a foothold in Europe, we are aiming to go across the Atlantic to North America and to US, which is the largest research organization in the research market in the world. Finally, we would also like to offer our fine technology to patients across the globe through a clinical product as well. So, as a summary, the product that we provide is giving recurring revenues. So for each sample, you need our particles. We are riding the digitization trend in healthcare, and we are fitting very well into that unstoppable train, I would say. Low barriers to profitability, a very lean organization, outsourcing everything that is not core to us. And we have a smart and unique technology, which is in many aspects superior than current technology. And by that, ladies and gentlemen, thank you very much.
Thank you, Matthias. We have a couple of questions for you here.
Great.
But yeah, go ahead and have some water.
Yeah.
Thanks. Let's start with the CE marking process, which is taking longer than expected and hoped for, of course. Why?
I think one of the things is that there were parts in the compliance testing when it comes to functional safety that had to be further in depth dealt with, and that's what's been occupying us over summer and up to now, I have to say. So now the instrument is back at NEMCO in Germany again to look at the updates that we had to do. So now they are actually checking that. So I would say the functional safety part within the compliance testing was the key driver for us to prolong the journey.
But you don't have an exact timeline for when this will be?
Yeah, we have an exact timeline with the stakeholders. NEMCO is one, and Optronic, which is our producer, and ourselves. So the timeline is now slightly into next year. So that's what the timeline looks like currently.
Right. Yeah. Let me see if there are any questions from the room. No, we will keep going with the app questions then.
Okay.
We have some questions on the technology. If you could give some examples in what user cases do you see that your technology really stands out?
Yeah, I would say when there are applications or tissue and situations where autofluorescence really is an issue, then we can completely mark that off because the physics of the wavelength of the technology, so it's really only showing the glowing nanoparticles and not the tissue around the area.
And is this also an alternative to molecular diagnostic testing?
No.
Short answer, very good.
Yeah.
Let's return to some money questions. What is the cost of your technology compared with existing technologies?
I would say very, very similar when it comes to scanners and so on. So more or less ballpark in comparing scanners. When it comes to the reagents, I think we in some aspects will be cheaper and compared to very simple stuff that doesn't provide you the value that we do. It will be more expensive, but it's a value pricing we are looking for, really, to be honest. Yeah.
Well, thank you so much, Matthias.
Absolutely. Thank you very much.
And now there's actually a change to the program. We will actually hear from Malin Bornstein, the CEO of Reccan Diagnostics. Welcome, Malin.
Thank you. Thank you very much. It's great to be here with all of you. I am Malin Bornstein, the CEO of Reccan, and I'm here to present the progress that we are making within the field of pancreatic cancer diagnostics. 500,000 patients are diagnosed with pancreatic cancer annually, and almost as many die from the disease. The prognosis at the time of diagnosis is very poor. Less than 10% in US and Europe survive more than 5 years. Early diagnosis is crucial for survival, but early symptoms are not conclusive, and most patients are diagnosed when it is already too late for resection and for curative treatment. And this is something that's not been changing over the past many, many years. There are well-defined risk groups, but no efficient means of screening. So there are imaging technologies that are labor-intensive and expensive and not suitable for testing at scale. Blood samples are suitable for testing at scale, but CA19-9 is about as old as I am, and it's missing up to 15% of the actual cancer cases. New technologies based on DNA are even worse, missing even more of the patients that need a diagnosis. So this is a clear unmet need, and it's also clearly stated in the national guidelines for pancreatic cancer. As an example, the patient group that has a defined hereditary or genetically increased risk of developing pancreatic cancer, they are supposed, according to national guidelines all over the Western world, except for Spain for some reason, supposed to be followed up several times per year. But the very same guidelines are also stating that efficient means of follow-up is lacking. New onset diabetes at an age above 50 is known to be connected with an increased risk of developing pancreatic cancer. That patient group is left without any follow-up at all, because of the lack of an efficient testing. Based on only these 3 patient groups, the serviceable market in US and Europe alone is 4.3 billion euros. In general, there's a large potential in early diagnosis of cancer, naturally. And this has led to vast investments. And the investments are showing progress for most cancer forms, but pancreatic cancer is not one of them, though it is becoming the second most common cause of cancer-related deaths. So all of these multi-cancer detection tests are going to have to be accompanied with a different test for pancreatic cancer if they want to state general early detection capabilities. We are in the final stages of developing such a test. We are working with 5 patented biomarkers specific to pancreatic cancer. And we are making sure that our solution fits to current procedures, current clinical procedures. And they are being developed to be run on already installed base of analyzers. The ones that are closest to us right now are here in Lund, There's install base in Malmö and Copenhagen. It's quite a common analyzer. Thanks to COVID, we all know that there's no such thing as a test that will never ever give a false positive answer and never ever miss a patient. If there were such test, that would have an area under curve of 1. Our test comes close. We have in repeated testing, when we look at comparing pancreatic cancer patients with healthy blood donors, reach an area under curve of 98.4 to 99.6. So it is fairly good. And as soon as we've finalized the development of our test, we are going to put our test to the test by analyzing a large population of already biobanked and collected samples to try all of these different relevant indications. That data is going to be used for finalization of our technical files. And then from then on, we are looking into prospective studies, how is our test performing in a real clinical setting, and then the launch, of course, into the areas where we have been granted regulatory approval. A very, very important step in launching this product is to get acceptance into the guidelines. We have seen, looking historically in the changes that have been made to pancreatic cancer guidelines, that inclusion into one guideline, inclusion into one guideline for one indication, has made possible a wider implementation, both when it comes to geography as well as indication-wise. We are, through our founders, very well connected with the specialists in the field, the oncologists and the surgeons. And we are well connected all over the world, specifically so in the Nordic region. And that is why we are planning on starting out with the Nordic region. There are 20 specialist hubs in the Nordic region. And we are, through our founders, connected with all of them. And some of them are going to be part of our prospective studies that we are planning right now, and in the work of getting the guidelines updated, because it is these professionals that are responsible for the update of the guidelines. The third step of our launch in the Nordic market, then we are moving outside of these specialist hubs, targeting a larger population of specialists that are getting into contact with pancreatic cancer patients. And we are also broadening the indications. So moving away from only hereditary and early symptoms patients. That is heavily interlinked with our next generation projects. We are already now looking into the markets that we have in our test to see how well they can also predict the outcome for a pancreatic cancer patient. And we will, as soon as our test is finalized, look into more of treatment and predictions of treatment. That's also an area that really needs to be taken care of. We know for a fact that about 1/3 of the patients who receive chemo today actually profit from it. So there's a lot of unnecessary cost and unnecessary suffering that we hope that our test is also going to be able to limit. We are also in the developed state of a lateral flow test that would be perfect to put at diabetic care centers for the diagnosis of new onset diabetes patients. So to recap really fast, we are almost done with the development of a test that is faster, cheaper, and more accurate to what is actually available on the market today. And we have a pipeline of new projects within the same clinical area. We are raising 10 million Swedish kronor right now. A large part of that has already been signed for. That is going to take us through the finalization of the development and the finalization of our clinical files. For the prognostic studies and also for the launch, we are going to raise, and next year, additional 20 million Swedish kronor. thank you. Now I have 30 seconds remaining. I'm going to have to talk about most important part of this. It's the team. Between us, we've got 50 years experience of surgery and research on pancreatic cancer. We have 150 years experience on development, production, marketing, launch of life science products. And we are also supported by experts on market access within the very specific field of pancreatic cancer, as well as IP. All of us have a strong sense of urgency. We believe that our test is going to start saving lives the very first day it becomes available on the market. Thank you all for listening. I hope that I've been able to transfer some of that sense of urgency also to you.
Thank you, Margaret. We have a couple of questions here that's come in through our app, and I should remind everyone, or maybe tell you for the first time if you're new, that on page 3 in the program there's a QR code that you could scan, and then you can send questions in, and I will read them out. So, you talked about pancreatic cancer here. How come that's the first cancer form that you're testing it in?
Very relevant question. So this was a pancreatic cancer project from the start. We are founded by surgeons that are active in that field, and we are founded from their frustration of not being able to help their patients. So they are following up on hereditary patients. There's no efficient means of screening, so there will be an imaging going through once a year. And that imaging is going to look fine every year until it doesn't, and then it's too late. So that's the foundation of the company.
But it could be used for other types of cancer as well?
I don't predict that it will be possible to use. It will be possible to use for differentiation from other cancer forms, so it will be relevant for other cancer forms as well. But it's not going to be useful in other cancer forms. And that's also why we're focusing our next generation projects on an increased usage within pancreatic cancer, because that's where we have our expertise.
So how does the competition look like within this area?
Yes. There's been a lot of money invested, both in general early diagnostics and in pancreatic cancer diagnostics. But so far with very little success. We were really hoping for one of our competitors to make it in the US, because that would have been beneficial, we think, also for our solution. But currently we don't have, we have general early detection companies, but they are not sensitive enough for pancreatic cancer. And currently there are other products in development, but nothing available. And we have not seen higher sensitivity in any other setting than what we seem to be able to provide from the data that we have gathered so far.
You mentioned here that the development within the area has been quite slow, and again, you were talking here about it's difficult to get tests that are sensitive enough. Why is it so difficult?
Pancreatic cancer is a difficult cancer form. So it's progressing faster than most other cancer forms. It's metastatic faster than other cancer forms, so it needs to be found earlier. That's one of the issues. And then it's also, so that's why, for instance, cell-free DNA isn't working very well. And then it's been more focused on more common cancer forms, where we've also seen a lot of success. And therefore, pancreatic cancer, but due to the aging population and also to the success in treating other cancer forms, that's the reason why pancreatic cancer is now becoming the second most deadly form of cancer, predicted to be in a couple of years' time.
And as a final question then, we'll talk about strategy. Why have you chosen the EU first and the US second and not the other way around?
Super good question. So when we decided on that strategy, we were hoping to be able to follow the strategy of being a fast follower. So that is something we are looking into that strategy. And secondly, we are connected all over the world through our founders, but we are exceptionally well connected within the Nordics. And we're really seeing that inclusion into one guideline is going to be beneficial globally, and that is the reason.
Thank you so much, Malin.
Thank you.
Next up, we have Cereno Scientific and CEO Sten Sørensen. Welcome, Sten, sorry.
Common mistake.
Yeah.
It's okay. Hi, everyone. It's good to be here. Always good to be at BioStock, both in interviews and at events like this. So I'm the CEO of Cereno Scientific, and we are pioneering new drugs for orphan and common cardiovascular diseases. I hope you'll find the presentation interesting. So our main program, our lead program, is trying to solve and provide value for patients with pulmonary arterial hypertension, very deadly disease, 7.5 years life expectancy in mean, mainly hurting females, but also males. So 7.5 years expectancy, and the drugs available today can't solve the problem. They're just symptomatic. Here needs to be agents that can remodel the structure in the vasculars. Now, cardiovascular is the most common cause of death for humans on the planet, and 80% of that is due to thrombotic events and causing damage to the major organs, so the brain and heart, so stroke and myocardial infarction. That's the second focus of our pioneering event. Now, the drugs available there, it's a major area, of course, but they are not effective enough. And the reason they're not effective enough, one of the reasons is that they're quite dangerous to use. 3 out of 100 die of bleedings caused by the drugs used in clinical material. 25 out of 100 get some kind of bleeding. That's a lot. So we aim to develop effective drugs that doesn't cause this bleeding risk. Now, we are pioneering, we believe. We have strong leadership, some of the best minds in cardiovascular research around the globe. And we are connected with both University of Michigan in the US, the largest university in the States, and their research. And also, we are doing a collaborative work together with Abbott, one of the giant medtechs, in our lead program in PH. We have 3 programs in development, 2 in HDAC inhibition, epigenetic modulation with HDAC inhibition. It's a new thing in cardiovascular disease. And we have a 3rd program, a prostacyclin agonist based on an androgenic lipid developed by my colleague instead at University of Michigan, which we have licensed for global rights. So we believe we are uniquely positioned to do something really good here. Our lead program is in pulmonary arterial hypertension. We are in phase 2, a trial pursued in the US at around 10 centers, 9 centers currently, and we are aiming to complete that by Q1 next year. This is the program we're doing together with Abbott and their groundbreaking technology implantable device to measure the pulmonary arterial pressure, CardioMEMS. I'll get back to that. Our second program is CSO14. It's soon completing phase 1 and moving into the clinic next year. So we're going to file an IND for moving into our phase 1 study. The third program is, as I mentioned, a prostacyclin agonist. That's in preclinical work. Now, looking at PH, there's a great unmet need, as I mentioned, and the market is around growing towards 11 billion, growing 6% healthy growth. But the need for new drugs is very strong, and I'll get back to why. So we have in CS1 a drug that has a multi-efficacy capacity as documented in preclinical work. So we have seen pulmonary pressure reduction, reverse remodeling on the structures of the cardiovascular and the heart. And we have antifibrotic, anti-inflammatory, and also antithrombotic capacity with this drug, all documented in preclinical work. And this fits very well with the disease pathophysiology in PH. And we are studying these effects in our trial in phase 2. The trial is safety and tolerance as primary objective, but we are studying a number of parameters, including structure, etc. The drug we're using is an old drug, VPA, used for prevention of epilepsy. So it's been in man for 60 years approximately. So it has very good safety and tolerability, and we didn't see any issues in our phase 1 study. We got an orphan drug designation by FDA, so we have 7 years exclusivity once approved and marketed. We have a strong patent portfolio, we believe, so 3 patent families, and we have documented or pursued that in 21 countries worldwide. So this is the study. We're studying 3 different dosages in these patients with pulmonary arterial hypertension, and they're being studied with Abbott's implantable device that's implanted prior to the study is randomized, or the patients are randomized into these different dosages. They're being followed 12 weeks, and that's the end of the study, and there's a 2 weeks follow-up after the therapy has been discontinued. The numbers here on the progress of the study is from August 23, and we'll come back with an update later to the market. Now, this summer, we really got some remarkable news about the study. An investigator in the study reported back after the patient had completed clinical findings out of the study, which we, when we got to know this, said we have to release this information. And so in this patient, 51-year female, been on drugs for 3 years, therapies, 3 different therapies, well established, and was added CS1, our drug, and we saw a 30% reduction of mean pulmonary arterial pressure. We saw an increase of 20% approximately on cardiac output, and a decrease of 43% on pulmonary resistance. So this is a remarkable improvement in a patient. And she moved from functional class 2 to functional class 1. In functional class 2, you're very impacted symptomatically by the disease. In functional class 1, you basically have your diagnosis. You don't feel that much from your disease. A number of other parameters were improved. So this gives us a very strong hope that we will be able to do something useful in this patient group. This patient had finished the study and was on the strongest dose of the 3 in the study. These findings prompted a data quality control initiative by our company and clinical steering committee, where we wanted to secure that the data transfer from CardioMEMS on efficacy parameters were optimal before we get to the end of the study in Q1 on all patients. So, and we also wanted to make sure that we had adherence to protocol. And so we did that, initiated that the 15th of September, and in that review was 16 patients out of the study. And these are the findings, so they were just communicated about 2 weeks ago. So the good thing we found here, of course, was that there were no concerning issues with the digital data transfer or a patient-physician protocol adherence. So that's key. But the other things that we saw gives us more hope of a positive outcome when we have the top line at the end of the study. It's that several patients in reduction of area under the curve, as measured with CardioMEMS on mean pulmonary pressure, had same or better reduction of pulmonary pressure than the patient case. So this indicates a clinically meaningful reduction of pulmonary arterial pressure in this study on top of standard of care drug therapy. In addition, we saw that 60% of the patients reviewed in this analysis had a sustained reduction of mean pulmonary arterial pressure. We also saw that the pattern of reduction of MPAP, mean pulmonary arterial pressure, was compatible with the dose response. And we don't know the dose, so the study is blinded for dose. This analysis was blinded for dose, but we we will see at the end of the study if we have a dose-response curve. We also saw an early onset of impact of CS1, so already after 3 minutes. And we also saw sustained efficacy after 2 weeks when the drug had been discontinued. So we are very much looking forward to the end of the study and the top-line results now. So we have these 3 drugs in development, and 2 of them have shown very interesting impact on thrombosis prevention. And as you know, thrombosis prevention is this major market is going to approach 43 billion by 2030. And we have seen efficacy on both our compounds CS014 and CS585 in very translatable mouse models, cremaster models, and without increasing risk of bleed. So this gives us very much hope in that area. So I would just mention that this portfolio that we have is now approaching interest to do business development deals or exits to pharma partners in the world, I think. So we are gearing up our activities with the portfolio, both for PH, the HDAC portfolio per se, and for thrombosis prevention. And the deal that was made recently, well, 2 years ago, but it's a benchmark deal in PH. Merck bought the company for $11.5 billion when they had finished phase 2 and were starting phase 3. So that's a very good benchmark. We believe that our product could beat sotatercept when we get to the market. Thank you.
Thank you, Sten. We have quite a few questions here, mainly concerning the results. So what do these results, what would you say they mean in terms of market potential?
Well, you know, we are a bit away from the market. So for a biotech, as you know, there are 2 markets. One is the deal market and one is the actual pharma market if you bring the drug to the market. Most of the time, pharmas get into a development deal or are bought out before the drug gets to the market. When it comes to orphan diseases, you could actually pursue your launch and marketing yourself. And we are keeping that door open for now. But of course, if our clinical data for CS1 would look as the patient case in the spring next year, I think we would be an attractive partner to do a deal with. So, and currently it looks actually better than sotatercept, both from a delivery oral, from side effect profile and efficacy profile. We'll see.
Have you had any comments from external researchers or industry people about this patient case that you presented?
Yeah, I think you know, if anyone here wants to listen more to we had a capital markets day it was held on the 30th of August, where we had Abbott, the head of chief medical officer for Abbott, presenting CardioMEMS. We had our principal investigator Raymond Benza presenting the trial and also the patient case. So I think from that perspective, but that's of course people involved in our effort. We have dialogues with potential interests, and we've had that for a while.
If we look ahead to a potential phase 3 studies, have you had any thoughts about potential primary, secondary endpoints for such a study?
I think you know what people really wants to see is the remodeling effect that we seem to see even in the patient case here. And currently, though, FDA is approving drugs on 6-minute walk distance, which is a physical ability. And I think until they change that, we'll have to have that in our data set when we go to market. But we will have a dialogue with FDA about our study and the data. In fact, FDA's Drug and Tech Division had a meeting about our study and approved to put CardioMEMS in the patients and CS1 in the patients to study them both, because this is the first study done this way ever. So you're able to follow the patient every day because CardioMEMS sends signals to computer. So we will have that dialogue with FDA and discuss the protocol when we are done.
Well, thank you so much, Sten.
Thank you. Appreciate it.
And our next speaker is Mia Bondin, the CEO of Synartro. Welcome, Mia.
Thank you so much. I think you've been sitting too long. Please stand up, everyone, just quickly. And then sit down again. So I'm Mia Bondin. I come from a company called Synartro, and today we will talk about a disease called osteoarthritis and the solution for many of the patients affected by that disease, and why this exercise besides keeping you awake. This is a very common exercise you have to do when you have osteoarthritis. Stand up, sit down, stand up, sit down. You can do it again later. So who are Synartro? We're a company developing local injections, and the aim of the local injections is to provide a longer efficacy for the active drug connected to our drug delivery technology. It's also aim of taking the drugs that have systemic side effects, and by injecting them locally, we can avoid many of the side effects. So our lead program is called SYN321. It's for the indication symptomatic knee osteoarthritis. And what we've done so far is we've shown initially that we have a sustained release within the knee joint, up to 10 times longer than for free diclofenac in the knee joint. And diclofenac, as you know, is the substance in Voltaren. This is the most potent anti-inflammatory drug for osteoarthritis treatment. We have patent protection starting in 2020. I will come back to that later as well. And we are now ready to go to clinic, straight into humans, patients with osteoarthritis. So let's talk a bit more about this disease. So we've been talking previously about diseases that can kill you. This will not kill you, but there is no cure for the disease, and many people are affected from a very young age. And it will continue and progress throughout your years, maybe those years when you want to be most active. So you want to treat these patients. It affects more than 500 people globally. And half of these patients getting treatment will not be pain-free. So they will have limitations to how they can move, what kind of activities they can do, how they sleep, because the pain is usually quite harsh in the nights. And this is something we want to change. The market is huge. It's projected to be, in 2030, $15.8 billion US. And why is this? Well, obviously the aging population, but also increasing number of sports injuries, etc. So let's have a look at the drug delivery platform and our molecule. This is a local injection. It's diclofenac, which is covalently bonded to a hyaluronan molecule by a unique linker. So you can see the blue part is the hyaluronan molecule. This is a big molecule, which is good because that makes it stay inside the joint. The linker itself doesn't really have any effect. It's just bonding the two substances together. And then diclofenac, which is a small molecule. So if you would inject that into the joint, it would immediately, or within a day or two, disappear from the joint. So you want it to stay there and provide its anti-inflammatory effect. So what happens when inside the joint is that through the natural substances in the synovial fluid, the bonds between diclofenac and the linker will break. So diclofenac will be gradually released for a long period of time. What is also important when you inject something into the joint is that you don't inject something that's not known to the body, because then you will have reactions. And this is what's good here, because also the hyaluronan, which is naturally present in the knee joint, will actually also release, providing viscosupplementation. This is just an additive effect, but the active substance here is going to be diclofenac. So let's have a look at the initial horse study that we did. We compared SYN-321, where everything is connected, to just injecting diclofenac and hyaluronan. So what happened? If you look to the left, you have the yellow-orange lines. This is the free diclofenac measured in the synovial fluid. So within 2 days, all the diclofenac was out of the joint, so no more effect. If you compare that to SYN-321, you could find diclofenac in the joint for 14 days, and this is when we stopped measuring, so we don't know how much longer it was actually there. So this is the proof of sustained release inside the joint. And you may think, okay, but 2 weeks, that's not so long. But if you compare this to, for example, corticosteroid injections, they are small molecules. You inject them, and you would at least expect like 6 to 8 weeks of duration of the pain relief from them. So the question is there, how long duration can you actually expect? Well, our expectations are that at least 3 months, but maybe up to 6 months of pain relief based on SYN-321. We have also completed the full preclinical and toxicology program for starting our phase 1/2A, which is planned to start next year in Q2. So why is there a need? You may think, okay, there are so many different products here, and you can't cure it anyway. So why is there a need? Well, as I mentioned, 50% of all patients getting treatments don't get enough pain relief. They still have issues. And if we have a look at the products that are on the market today, or the categories of product, they are all connected to different limitations. First of all, the opioids, maybe not so common here in Sweden to use, but in the US, for example, which is going to be the biggest target market for us, for SYN-321, is widely used. And of course, you get addicted to opioids. You want to avoid those. If we have a look at the corticosteroids that are injected, could give some pain relief for up to 6 to 8 weeks. But on the other hand, in osteoarthritis, the cartilage in the knee joint is broken down. And the effect of repeated use of corticosteroids are known to also break down the cartilage. So it's the opposite effect. This effect we don't see at all in the preclinical studies with SYN-321. And of course, oral treatments with anti-inflammatory drugs of different kinds, widely used. Even if you get the injection treatments, you can get them on a regular basis. But most people cannot tolerate these products. You get the side effects from the systemic treatment, from the GI tract or from cardiovascular, kidney, etc. And many people have other diseases, so they can't use these products. So finally, you have the pure hyaluronic acid injections. A little bit question how long duration you get from it. You get a sort of lubrication of the joint, which is good, but that is not enough usually. You need the anti-inflammatory effect as well. So there is clearly a void which needs to be filled. The market potential is huge. Our rough calculations give us, for the US and part of the EU market, $600 million, 5 years post-launch. And we are totally ready to start the first clinical trial next year. The patent side looks good. We have a protection for the base compound, where we can also attach other molecules and build new projects upon that. And of course, for SYN-321 as such. And we've also added some new applications regarding the formulation and sterilization. We can also add indications, of course, for SYN-321. And as mentioned, we can develop new projects with other active substances on the base molecule. And the team, of course, is highly important in this. I have one of my colleagues with me here today. And we also have Stefan Lomander as a senior advisor here from Lund University. and the board of directors also contributing with their very long expertise within the field. Thank you so much.
Thank you so much, Mia. Got a couple of questions for you here. Yeah, someone's asked who your main competitors are, but you did go through that, but could you just maybe sum that up and explain how you will position yourself within that landscape?
Yeah, of course. I would say that our direct competitors would be the other injectable products like pure hyaluronan injectables, but also corticosteroids. That would be where we would have the sort of easiest catch. But also, I would say, from the oral treatments.
So could this be considered a reformulation, what you're doing?
I wouldn't say so, because it's actually going to be as a new chemical entity when we register it. Because the different substances are bonded to each other, so it's not only mixed.
We also have a question about the financial status of the company, how well financed you are.
Well, as a private company doing clinical trials, I would say we always need more money.
It's a very honest answer.
Yes.
Could a long-term effect mask other knee injuries if we return to the clinical side of things?
That's a very good question, but I would believe, I mean, there are the majority is the benefits of having a pain relief, because then you can actually move and exercise your knee and your muscles, so you help with managing the disease.
And you did mention here that your drug delivery platform can be used for other indications. What are you looking at specifically, and how do you make those decisions?
We've just started that work, doing some preclinical testing. We've tested attaching antibiotics. We've tested other anti-inflammatory drugs. So that would be the easiest way. But we also have the possibility to do chemotherapy and adding that.
So that's an ongoing project?
Yes, it is.
It's an ongoing. Thank you. So why have you chosen this patient population to start with?
Well, the knee osteoarthritis is a huge market, of course. And I mean, it's 80% of the market of osteoarthritis, so it was very natural to start there.
And I guess the US is also the natural place to start?
Absolutely.
Yeah. And then will you go for EU as a second market, or were you only thinking the US for now?
We're planning to do it in parallel.
In parallel, right. Thank you. So as a final question, I wanted to ask you a little bit about what kind of feedback you've had from key opinion leaders and from the profession.
Yeah. From the discussions we've had with physicians in Europe, but also in the US, they say everything that's better than what we have is good. So please bring it to us, because there is a huge need for better products, and especially to avoid the side effects of what's there and the addiction side of opioids, for example.
Right. Thank you so much, Mia.
Thank you.
We will then turn to our 3-minute pitches again. And just to remind everyone, these are 3-minute pitches by unlisted companies with, shock horror, only one PowerPoint slide. So those are the rules. And first out is MyCural Therapeutics and CEO Alena Castell.
It was discovered already in the 80s. One of the main reasons why cancer cells divide uncontrollably, why they resist treatment, and why they spread throughout the body. This holy grail of cancer is a protein called MYC. You find the name of it in our name, MyCural, MYC. This has been a highly desired target for drug discovery for decades. Unfortunately, there is no anti-MYC therapy. And why is that? Well, as I said, decades of funding, efforts, and no one has actually really managed anti-MYC therapy. Although we knew already in the 80s we wanted it. So why is that? Well, MYC is a very difficult target, and common drug development methods cannot be applied to it. So methodologies and also instruments have been underdeveloped, and knowledge have been insufficient. So what do we do about that? Well, we know how to do it now. For 20 years, cancer researchers from 33 universities in Sweden have dedicated their time to try to understand this main driver of cancer. And together with MyCural, we have built a platform with methods that are unique to be able to identify and validate potential candidate drugs that can be developed into an anti-MYC therapy. Today, MyCural Therapeutics has potential candidate drugs that we want to develop into this therapy. These potential drugs are efficient, they are selective, they inhibit tumor cells within a wide range of cancer, and they are regressing tumors in animals. So MyCural is now embarking on a program where we will bring forward a candidate drug that we will develop into an anti-MYC therapy, the first time in history. So, as you understand, this is a huge market with immense revenues. And with that, I would like to say, be brave and join us on the mission to cure cancer.
Thank you, Elena. Our second pitch is from Uman Sense and CEO Cecilia Belfrage.
Hi everyone, I'm Cecilia. 1 in 4 will suffer a stroke, the leading cause of disability in adults, and the cost for the society is huge. 20 years ago, the stroke patient was last in line in the emergency room. There was nothing to do. They would only go to rehabilitation. But today, healthcare can cure stroke. With a small incision in the groin, they go all the way up through the blood vessels to the occlusion in the brain and remove the clot. So the blood flow is restored. But a large majority of those patients arrive in hospital too late. Either they don't understand they suffer a stroke, or they can't alert due to the stroke. And this is why we have developed Stroke Alarm, alerting contacts in case of stroke symptoms. The basic principle behind Stroke Alarm is that 83 to 90% of all stroke patients suffer arm paralysis. That's what we are looking for. The system consists of 2 bracelets, 1 on each arm, and they are paired to the patient's own smartphone. If the system gets sign of stroke symptoms, then contacts are alerted so that the patient can get to hospital in time for treatment. And the system, the Stroke Alarm, is a medical device. It is CE marked as Class 2A. And so who should use this? Well, there are many risk groups, there are many risk factors for stroke, but there are also some well-defined risk groups with very high risk. If you had a TIA or a stroke, you have about 9 to 20% risk of having a stroke during the first half year. If you have atrial fibrillation, you have about 3 to 5% risk every year for the rest of your life. Many, many, many patients. We want to, Stroke Alarm will be sold to healthcare providers or insurance companies for high-risk patients through partners and distributors. And since Stroke Alarm has market clearance in Europe, our focus now is to launch Stroke Alarm in Europe. And now we're raising 9 million Swedish crowns. About 6 of them are signed for. And it's for preparing the launch in Europe together with a partner by the end of 2024. So if you want to help us cure stroke, please reach out. Thank you.
Thank you, Cecilia. And our last pitch is from Nygen Analytics and CEO Parashar Daplo. Welcome.
Thank you. Picture a world where you can discover drugs faster, you can do an early diagnosis of disease, and then you can predict the therapeutic response with much ease. I'm Parashar Thapola. I'm the co-founder and CEO of Nygen Analytics. And what we are doing is that we are on a mission to make this world, this is what you pictured now, a reality by using single-cell genomics technology. Single-cell genomics is like looking into the secret lives of cells, looking at what they're doing, and generating this rich information that we were not getting before with conventional genomics techniques. Today, more than 8,000 research labs across the world are using this technology and trying to understand the various disease models in mice and get better insights into the human samples that they have obtained. Single-cell genomics is what is going to be the future of precision medicine, and we truly believe so. As these researchers are generating a large amount of data, what they're struggling is with data analysis. With decreasing sequencing cost, generating large-scale data is not a problem, but analyzing it is, because people do not have access to the analysts, they don't have access to this expensive compute requirements that's needed to analyze the data. What we have done as a spin-off from Lund University is developed a completely new cloud architecture to analyze single-cell genomics data at 10 times lower price. What this has led us to do is that develop this solution and provide it to the genomics researchers across the world. And right now, since our launch in December last year, we are seeing 35% month-over-month growth of our user base. We have signed deals with Lund University and University College London and have already booked our first 100,000 crowns in revenues. What we are doing now is trying to bring our product out to more researchers, trying to develop further data products built on our core offering. We are a team of 7 people based here in Medicon Village, and we are looking for our investors for our next round, our seed round. And if you're passionate about genomics or just generally want to know more about single-cell genomics, please feel free to talk to me.
Thank you, Parashar. Thank you.